Aging is a complex and dynamic process ongoing with time, which includes the biological, psychological, and social aspects. Of them, the biological aging is the most studied one, featured by progressive and irreversible changes in metabolism and physicochemical properties of cells. These accumulated changes result in diminished cell self-regeneration, increased vulnerability to stress, structural and functional changes of organs, and the development of myriad diseases and disorders. Extensive efforts have been made to understand the biology of aging and its regulation, but limited breakthroughs have been made. In 2013, Carlos et al. Proposed the 9 hallmarks of aging based on 3 criteria: the time-dependent manifestation of alterations accompanying the aging process, the possibility to accelerate aging by experimentally accentuating the hallmark, and the opportunity to decelerate, halt, or reverse aging by therapeutic interventions on the hallmark. After that, several studies summarized the hallmarks of different types of cells and cellular senescence. Recently, Tomas et al. Formulated new hallmarks of aging, which include compromised autophagy, microbiome disturbance, altered mechanical properties, splicing dysregulation, and inflammation. In 2021, Carlos et al. Proposed the hallmarks of health, emphasizing the importance of spatial compartmentalization, maintenance of homeostasis over time, and adequate responses to stress in maintaining health and longevity. In 2023, Carlos et al. Updated the hallmarks of aging and added another 3 hallmarks. After comparing the healthy, aging, and diseased state of cells, we found that mitochondrial biology, including its structure, biogenesis, function, localization, homeostasis, mitophagy, and genome stability, plays a key role in aging and aging-related diseases. Mitochondria are responsible for cell energy supply and involved in cellular bioenergetics, genomic stability, reactive oxygen species, ROS, production, ion homeostasis, active stress responses, and cell fate determination. During aging process, internal and external stresses cause a decline in mitochondrial quality and activity, featured by abnormal morphology, accumulated mitochondrial DNA, mtDNA, mutations, loss of mitochondrial membrane potential, MMP, impaired mitogenesis, and decreased mitocytosis and mitophagy. These changes are inducers of age-related inflammation, also referred to as inflammaging, cellular senescence, and cell apoptosis. Besides, mitochondrial dysfunction has been associated with a wide range of age-related diseases, such as Alzheimer's disease, AD, Parkinson's disease, PD, osteoarthritis, OA, metabolic diseases, and cardiovascular diseases. Several anti-aging drugs targeting mitochondria and mitophagy have shown promising results in restoring mitochondrial function, increasing health span, and mitigating degenerative diseases. Mitochondrial transfer and mitochondrial transplantation are concepts that have attained ample attention in recent years. It makes sense to consider these 2 subjects together since they cover common topics such as mitochondrial donor selection, the mechanism by which mitochondria enter recipient cells, and the effects that mitochondria exert. As described in this review, the exploration of spontaneous mitochondrial transfer provides a theoretical basis for the future treatment of diseases by mitochondrial transplantation. Although mitochondrial transfer/transplantation mechanisms are currently not well understood, these processes still show great therapeutic potential. Thus, extensive and normative experiments should be performed to understand the molecular and cellular mechanisms of mitochondrial transfer/transplantation and to demonstrate their efficacy, which will serve as the foundation for future clinical trials. Mitochondrial transplantation therapy presents a new paradigm of therapeutic intervention, which involves the supplementation of healthy mitochondria to replace the damaged mitochondria for the treatment of neurological disorders. Prior studies demonstrated that the supplementation of healthy donor mitochondria to damaged neurons promotes neuronal viability, activity, and neurite growth and has been shown to provide benefits for neural and extraneural diseases. In this review, we discuss the significance of mitochondria and summarize an overview of the recent advances and development of mitochondrial transplantation therapy in neurodegenerative and neurovascular disorders, particularly Parkinson's disease, Alzheimer's disease, and stroke. The significance of mitochondrial transplantation therapy is emerging as they meet a critical need to develop a disease-modifying intervention for neurodegenerative and neurovascular disorders.